this over. Great. Okay. Great, thanks. Yeah, I was just going to test that. Okay, great.
We have a good crowd today. They're enjoying the refreshments are delicious. Okay, great. Avoid the bathroom.
Well, good afternoon. It's really a delight to welcome you to the 2016 Harrison and Lya Latta Endowed Professorship. Today, or this year, our honoree and speaker is James Versalovic from Texas Children's Hospital and Baylor College of Medicine. A word about the lectureship. This was established about 20 years ago by Harrison Latta, distinguished faculty member of our department, and his spouse, Lya Cordova Latta, who is a passionate and a passionate philanthropist in the Los Angeles community for a number of concerns of social, cultural, and medical interest. The purpose of the lectureship is the opportunity to bring to UCLA in Los Angeles the luminaries of the field of pathology, particularly those focused on innovation in translational and clinical medicine. And there couldn't be a better example of that than today's speaker. Dr. Versalovic is, as I said, is the pathologist-in-chief at Texas Children's Hospital and the director of the Texas Children's Microbiome Center. He's also the Milton Finegold professor and vice chair of pathology and immunology at Baylor College of Medicine. It is at that institution that he had his MD training and his Ph.D. He then went to have his clinical training at Massachusetts General Hospital and then to MIT at the Center for Comparative Medicine, where he did remarkable work on microbiology and host microbial interaction. After a period at Harvard, he was wooed back to Texas, where he rose through the ranks to become not only a leading mind at that major institution, but also in the fields of pathology, clinical microbiology, and microbiome research. He's on the clinical side. I would just note that he's the editor-in-chief of the Manual of Clinical Microbiology, which is a leading textbook and and thought-leading volume in that field. For more than 20 years, Dr. Versalovic has done remarkable work on translational microbiology. His work has included pathogen biology of organisms. He has done striking work on the structure and function of the microbiome and has been one of the important voices in minds in the development of that rapidly emerging field. And in that, examples of recent work which has gotten a lot of attention has been understanding microbial communities which are poorly understood but potentially of great importance in biology, such as the prenatal microbiome of the placenta. Perhaps his largest ongoing work has been to understand what are the products of the microbial community which affect the physiology of the host. And in that, his work on the production of bioactive molecules, particularly neurotransmitters of that category, I think has been formative in a fresh thinking about the molecular ecology of the microbiome with regard to the host. And so very much looking forward to, as we all are, to his talks today, and so glad to have you come to visit Los Angeles.
Thank you. Thanks, Dan. Well, my thanks to Dr. Braun and the department for hosting me today, and for Pira, too, to getting me around and visiting with several of you. And I really do appreciate this honor and the opportunity to give this very special lecture here at UCLA, the Harrison and Lya Cordova Latta Lecture. And so it's really my pleasure to describe and privilege to describe our work in the intestinal microbiome and how we can begin to think about these emerging concepts and applying them in this era of microbiome science and metagenomic medicine. I'll just mention one disclosure. We have received some support from BioGaia. They are in the probiotics arena, so I'll mention that. And so at the beginning, I want to highlight the fact that we're very fortunate in Houston, and I know in this community here at UCLA with Dr. Braun's own research efforts and leadership, you have an active microbiome science community that's growing and very dynamic here. And certainly in Houston, we've been fortunate to have several key faculty, and I'll be alluding to some of their work today, and certainly this has been a team effort. Tor Savage, our associate director, and with also leading light in C. difficile pathogenesis and GI inflammation. Emily Hollister, Tony Haig, who leads our microbial metabolomics proteomics unit, and several other faculty listed here who are working together with our staff to really think about how within our Department of Pathology at Texas Children's, how can we then begin to translate this knowledge to impact medicine. And so we clearly are confronted with a very diverse microbiome, and this has become old news already in 2016. During the past decade, we've certainly made plenty of progress in characterizing the various bacterial and other microbiological components of the human microbiome, developing animal models to explore this, and also to think beyond composition to function. And so this slide just nicely illustrates the complexity, the variety of players in the microbiome that we've considered in different body sites, and this was generated by Curtis Huttenhauer and colleagues at Harvard. Clearly a number of examples that are important in different body sites, each body site having a distinct signature of key organisms. We know there are 4 abundant phyla in humans, and certainly we've begun to understand which key players are exist within each phylum, keeping in mind that there are more than 50 known phyla in the environment if we consider the Earth's microbiome. And so there is a defined subset of high abundance and low abundance phyla. And of course, these are groups that contain many different species. The real challenge now is to get beyond the descriptions to action, really, in how we might be able to take this information, make it actionable for medicine and also to understand basic mechanisms of human physiology and disease. So I have three main topics today, and one is relatively crude and simple, but I think deserves mention at the beginning if we're going to talk about how we are applying microbiome science in medicine, and that is the example of Clostridium difficile, antibiotic-associated diarrhea colitis, and the radical reshaping of the gut microbiome with fecal microbiota transplantation. And I'll follow that with examples of microbial components of chronic disease with respect to irritable bowel syndrome and inflammatory bowel disease. So first, C. difficile. It's certainly, to some of you, may seem like quite an old topic and certainly preceded the era of microbiome science, which we can really trace back to 2005 and the Paris meeting when the term was really articulated clearly for the first time in a major international meeting. And of course, following that, the formation of MetaHit in Europe and the Human Microbiome Project in the United States in 2007, 2008. This organism was well known by then, characterized for decades. We knew we could culture and identify this organism since the late 1800s. We knew about the entity of pseudomembranous colitis since the 1930s. We knew through early efforts in anaerobic microbiology about Clostridium difficile, or the difficult one, and interestingly, in the colonic microbiota of newborns. It wasn't until the 1950s that there was an association and this notion of a disorder of microbial ecology, perhaps with the connection between antimicrobial therapy and pseudomembranous colitis. And then in the late 1970s, hard to believe it's been almost 40 years, that C. difficile toxins were identified as a cause of pseudomembranous colitis. So that key connection was made between a bacterial toxin, antimicrobial therapy, and a potentially life-threatening colitis. So we certainly are well aware throughout the United States in both adult medicine and pediatrics that C. difficile is thriving and has continued to increase in number in terms of cases responsible for hospitalizations in the first decade of the 21st century. The numbers are in, and it's quite dramatic, as you can see on this graph. And certainly in 2011, from this report in the New England Journal of Medicine, more than 500,000 infections in this country. So clearly a major pathogen. As we were beginning to understand the human microbiome, we were confronted with a major clinical phenomenon. And so the Clostridium difficile genome was published for the first time 10 years ago. It's the right time, I think, to just reflect on that. It's been just a decade since this publication in characterizing one isolate from a hospitalized patient from Zurich, Switzerland with severe pseudomembranous colitis, demonstrating the existence of large genes that could encode each toxin, and the fact that this genome is highly mobile with mobile genetic elements in mosaic, 11% of the genome comprised of mobile genetic elements. So clearly we could begin to understand the plasticity of these bacterial genomes in the gut already in 2006 as a number of gut bacterial genomes were being sequenced. Just before we began these high-throughput metagenomics efforts. And so other fundamental insights that were being articulated in the scientific literature as we began to characterize the metagenome in the intestine, the work of Vince Young and others. Vince is now currently at the University of Michigan, at the time at Michigan State, described the key message that the microbial communities in patients with recurrent C. difficile infection, that is, those patients failing 2 courses of antimicrobial therapy targeting C. difficile, those microbiomes were relatively limited in terms of microbial richness and diversity by sequencing the fecal microbiome compared to healthy controls and those with a single bout of treatable C. difficile infection. And so that pointed the way beyond detection of toxigenic C. difficile with molecular methods, which it's helpful to remind everyone that is really just about a decade old. The widespread application of molecular diagnostics to detect toxigenic C. difficile with sensitive assays that are widely available, and beyond that, to appreciating the fact that there were, despite the ready availability of rapid molecular diagnostics and antimicrobial agents to treat, we knew that we had a problem with recurrent C. difficile infection. We could begin to explain this phenomenon by understanding gut microbial composition beyond the presence or absence of toxigenic C. difficile. So that created a lot of interest around 2011, 2012. If you follow the sequence now, you begin to see why by 2013 there was a major pent-up demand for fecal microbiota transplantation, an approach that was pioneered by the early 1990s by Tom Borody, a gastroenterologist in Australia, and a few others as a possible treatment option for various GI disorders. And it became clear by early 2013 in this seminal paper in the New England Journal of Medicine by a group in the Netherlands, in the first prospective study published using FMT head-to-head with antimicrobial therapy for recurrent C. difficile, that fecal microbiota transplantation could easily outperform vancomycin therapy in this context of recurrent CDI. So attempting to treat first with metronidazole, failing, then exploring whether one could then treat with a second antimicrobial agent that we knew was effective, at least in a subset of cases, already established as a treatment option of choice for C. difficile, but failing miserably when compared to FMT. And I don't think that's an exaggeration, of about 30% efficacy versus 80+% with a single infusion of donor feces. This is important, I think, to just pause for a moment and note that this was simply 3 years ago, when we really had now a strong case for applying now an ecological approach, however crude, with stool as a mode to transfer, revamp, or transplant a microbiome into a diseased individual. So with a chronic GI infection, we could begin to appreciate that perhaps resetting this depleted microbiome and placing it in a different alternative state and applying ecological theory toward an understanding of the human microbiome and how we might begin to treat and cure patients, as articulated here by this review by David Relman, we could begin to see that FMT may have a place in medicine. And beyond FMT, could we begin to now develop microbial communities, mixtures of microbes in pills or tablets that could be used in the future to enhance or reset, basically reboot the microbiome into a healthier state. And so we've extended that into pediatric C. difficile. Now keep in mind, these 3 publications, and I'm taking just a few examples from the literature, followed this is 2014. In 2013, a key meeting in Bethesda, Maryland at the NIH, when a number of us were present there, investigators, physicians, scientists, along with the FDA and NIH, and patient advocates who were asking and begging, frankly, for the FDA to relent on the enforcement of the IND. We had just started to apply FMT in children earlier that year and were put on pause while the FDA reconsidered. And fortunately, through the efforts of the AGA and others, other advocacy groups, by June of 2013, the FDA backed off and said that the scientific literature, again, taking the evidence in hand, was enough to justify the use of FMT for recurrent C. difficile infection. And so then the pediatric studies followed the year after, demonstrating clearly that what worked for adults could work for kids as well. And we have applied that at Texas Children's, similar to other pediatric and adult centers, taking individuals who failed 2 rounds of antimicrobial therapy with metronidazole and then vancomycin to then apply FMT since June of 2013 without FDA IND approval. And so the field has flourished since then, and certainly there are many patients who have benefited from this approach. Of course, we've tried to steer away from self-directed therapy to doing this in a controlled medical setting to ensure that we had healthy donor stool that we could place by colonoscopy into the proximal large colon, and that if we did not see resolution of symptoms, our pediatric gastroenterology team would repeat the FMT with retention enema. If we did see resolution, we would still follow by 16S ribosomal RNA gene sequencing at 2 weeks and 2 months post-FMT to correlate symptom resolution with molecular findings, and then allow those patients to then simply move on with their lives. And this has been successful as an active collaboration as we've been banking donor stool in our microbiome center in pathology. We've had 18 patients to date, actually 19 as of yesterday, but we don't have the follow-up data on that case. These are children ages 2 to 16, and of course we are well aware of the active discussion about whether infants and toddlers truly are susceptible to C. difficile infection, but doing extensive clinical correlation with our GI group, we felt it was worthwhile to pursue these cases of recurrent CDI with documented colonization by toxigenic C. difficile. 13 of the 18 have yielded complete resolution of symptoms, again, linking, correlating at 2 months follow-up, and that's generally accepted as the follow-up period. 2 months, normal, or basically a healthy gut microbiome by 16S sequencing, and symptom-free. There are a few that did not resolve, and note that several of these had complicating factors such as ulcerative colitis or IBD. And I'll note that, again, we've had some success with IBD and C. difficile infection, but it's been mixed. And we have tried FMT under IND for ulcerative colitis, and frankly, it's failed. And I'll get back to that point. But for basically a depleted microbiome that's been overtaken by a pathogen such as C. difficile, this strategy of FMT has been effective overall in our patient population with recurrent C. difficile infections. So we are moving on thoughtfully, carefully selecting patients who may benefit from this therapy. But even in children, though the disease is less severe generally, it means that they are now free from symptoms and can move on without further regular consultation with a gastroenterologist. So it's made a real impact with our patients and their families. And as an example, pre-FMT, you see examples of a microbiome that's clearly aberrant by 16S sequencing, easy to see versus the donor prep, healthy young adults, and C. difficile in the same patient, I should say the 16S sequence in the same patient post-FMT for recurrent C. difficile infection. You see that normalizing, and I'll take this example to highlight the presence of relative abundance of Bacteroides and Rosaburia, as we would predict from the donor microbiome. So clearly we've been able to follow this and illustrate this several times, and I'm just showing one example on this slide. So that's the case for C. difficile and advancing metagenomic medicine, and I think clearly for this and other enteric infections, and we recognize that there are other causes of antimicrobial-associated colitis, such as Clostridium perfringens, and it may be also, we think, this will be an effective strategy for cases where the dysbiosis is severe and we have basically a dominant pathogen that needs to be flushed out by radically revamping the microbiome via FMT. That's not going to work, though, for other examples in gastroenterology and gastrointestinal pathology. And so clearly we have plenty of work to do in understanding the mechanisms of host microbial crosstalk, and we know that microbes and humans are working together to produce metabolites. They may be co-metabolites or metabolites produced by either microbes or mammalian cells, and then constituting a holometabolome. And this holometabolome concept has become very important as we think about how these areas of human biochemistry and medical microbiology or microbiome science begin to come together. And so in the context of C. difficile infection, we've been considering various metabolites too that may give us clues as to novel or new therapeutic interventions. that may also supplant FMT in the future. And that is, one example is GABA, or gamma-aminobutyric acid, this neurotransmitter produced by the decarboxylation of glutamate. And through Tor Savage's work, we've noted in the center that we see an enhancement or increase in GABA in patients with recurrent C. difficile infection. And I'm showing Tor's picture again to highlight the result of months of work in beginning to create the visual analytical platform to show the correlations between LC mass spectrometry data, metabolomics, and 16S ribosomal RNA gene profiling in patients. These are children with C. difficile infection. And I'll cut to the chase in the bottom left to highlight that we are beginning to identify metabolites that are firmly associated with one state or the other. In this case, GABA in the red square associated with cases of recurrent C. difficile infection and pre-FMT, as you see by the yellow triangles. And by contrast, bile acid metabolites, secondary bile acids like lithocolate, keep in mind that primary bile acids are metabolized by the gut microbes, and lithocolate appears to be associated then with healthy controls and the cases post-FMT that have complete symptom resolution. So it may be that we could follow specific metabolites in the future as another strategy to examine these patients after a therapeutic intervention. So we're beginning now to think about clinical chemistry in new ways, in this case using stool, but we could think about other body compartments such as urine specimens as well. So that's the introduction into the metabolomics part of this talk and considering microbial metabolites. And I'll move now into irritable bowel syndrome and recurrent abdominal pain, and to highlight that none of this would be possible without this collaboration with Rob Shulman, a leader in our pediatric gastroenterology section with respect to functional GI disorders. We've also been collaborating more recently with Emran Meyer here at UCLA, and certainly this is a place here at UCLA where there is plenty of expertise in gut-brain microbiome axis. Our own work is focused on children. We know recurrent abdominal pain and IBS are major problems for both children and adults. And again, classified as a functional disorder, we're trying to understand, however, mechanisms of disease and getting beyond simply functional abdominal pain to irritable bowel syndrome, which is recurrent abdominal pain with either constipation, diarrhea, or both. Chronic GI disorder, and 5 years, it's hard to believe it's been, I guess, less than 5 years, but more than 4 years ago, we published in Gastroenterology one of 2 back-to-back papers describing microbiome signatures in children with irritable bowel syndrome. These are pre-adolescent children, and a separate paper from the Netherlands and Willem de Vos's group showing some overlapping features and distinct signatures in the microbiota of adults with irritable bowel syndrome from Western Europe. And we had a few common signals, one being the enrichment or greater relative abundance of gamma proteobacteria, a group including a variety of gram-negatives, commonly recognized gram-negative enteric pathogens, and perhaps lipopolysaccharide and other signals from these bacteria that we know are potent inducers of inflammatory cytokines. There may be some low-level inflammation contributing to IBS, perhaps some other features as well. So we began by looking simply at microbial composition using the standard methods of 16S ribosomal RNA gene sequencing, but we're now trying to get beyond that to examine what happens with a therapeutic dietary intervention and whether we then could follow certain microbial signatures or metabolites. And as mentioned earlier by Dr. Braun, we've been actively exploring a variety of microbial metabolites, including neurotransmitters such as GABA, I just mentioned, in the gut with respect to C. difficile pathogenesis, and perhaps, as scientists such as Elaine Shao here have shown, the induction of serotonin production by enteroendocrine cells, a variety of compounds that may be of interest. And so we're now beginning to explore the function of the microbiome and its relevance to chronic GI disorders. So we get beyond infection to microbial components of chronic disease. And in this case, we were very interested with Rob Shulman and Bruno Ciampatazzi to study the features of the gut microbiome that may help predict which patients may respond to a dietary intervention. can we begin to link, again, diagnostics and therapeutics using metagenomic medicine? And so we've been following and actively participating in the development of FODMAP diets, low versus standard, so low fructan, oligosaccharide, disaccharide, monosaccharide, and polyol carbohydrate groups that are considered to be a possible factor in at least a subset of IBS cases by providing malabsorbed dietary carbohydrates that lead to a high osmotic load, fermentable substrates within the microbiome that may lead and contribute to gas production in the intestine, increase in fluid, luminal distension, and then various symptoms associated with recurrent abdominal pain, and more specifically irritable bowel syndrome. So within adult and pediatric gastroenterology, there's been activity within the past several years in applying low FODMAP diets to try and remove this substrate and see whether certain patients might respond by symptomatic resolution. And so our initial paper in this area, admittedly with a small number of patients, was published in 2014, where we began now to link individual microbial taxa such as Sporobacter and Subdoli granulum, as you see here, with responders versus non-responders to a low FODMAP intervention. And in contrast, well, I should say in addition to microbial taxa by 16S sequencing in this beehive plot, we also were able to link various microbial metabolites or co-metabolites that may be signals of, that may help us identify responders pre-intervention. So these are samples taken before the low FODMAP intervention, and so you can see that a variety of amino acid and bile acid metabolites were linked with response to the low FODMAP diet, other aspects such as lipid metabolites linked to non-responders. So we begin to see possibly some microbial biomarkers of interest. And we took this further in the subsequent year, less than a year ago, published this manuscript together with the same group at Texas Children's randomized clinical trial examining gut microbiome biomarkers associated with clinical response to a low FODMAP diet in children with irritable bowel syndrome. And so in this, we've got basically 2 days on the low FODMAP diet, a 5-day washout period, and then a crossover design, 2 days again with the other diet. And you can see here comparison of low FODMAP plus fructans, low FODMAP plus maltodextrans. So basically this is your low FODMAP intervention. And so what we found was that we've just begun to identify key microbial taxa that may be relevant, that help explain which patients may respond to a low FODMAP diet. And you see various taxa within the genera Bacteroides, Ruminococciaceae, and Faecalibacterium and Doria, well known for housing carbohydrate-active enzymes. And perhaps these organisms may then indicate the ability then to respond to a low FODMAP diet. And additionally, we're beginning to identify pathways involved in carbohydrate metabolism, such as LACI regulators, transcriptional repressors, and starch phosphorylases that may be enriched in individuals who respond to IBS. So there may be features of microbial carbohydrate metabolism that may be relevant in these responders pre-intervention. Our key challenge is to identify those key subgroups. And in contrast, we found a particular microbial taxon, Thericibacter, that seems to be a recurrent signal in those children that did not respond or failed to respond to a low FODMAP diet. So this genus, interestingly, is a very different pattern of carbohydrate fermentation that you see listed there, thriving on fermentation of maltose and 5-ketogluconate. And perhaps these organisms are just resistant to that carbohydrate intervention and may also affect the overall microbial community in a way that might tilt an individual towards resistance to this specific dietary intervention. So I bring these examples, though they are preliminary, to begin to point the way towards possible microbes or microbial pathway features that may be useful in stratifying patients and to then enable us to couple patients with specific dietary interventions. So moving on to the last segment of this talk, the third and last area, which is in chronic intestinal inflammation, again, chronic disease, and then possible implications for inflammation-associated colorectal cancer. So beginning to tie these two together, since we do have current funding from the National Cancer Institute and plenty of interest in that connection. So histidine, I've talked a little bit about carbohydrate metabolism. I've mentioned GABA and glutamate metabolism. And now we'll delve deeply into a particular aspect of amino acid metabolism by the microbiome. L-histidine is an amino acid considered essential early in development of humans and other mammals, certainly an important amino acid that may be modified in different ways and metabolized to form different compounds that may suppress inflammation and carcinogenesis in a variety of animal models. Compounds such as carnosine, the dipeptide that includes L-histidine, urocanate, that's considered to be important in the skin, inflammation in the skin, and histamine, well known to many, and certainly an old molecule by any standard in biochemistry, because we've known about histamine since the early 20th century. This story is really a century old. But what we've been studying for the past several years is the metabolism of L-histidine to histamine by the microbiome, and then the communication between microbe and mammalian host via histamine to histamine receptors. And so we'll be focusing on this now for the rest of this seminar. With Jennifer Spindler, a faculty member in our Microbiome Center, and others, we've been characterizing a variety of intestinal and milk-derived, human milk-derived lactobacilli. And these lactobacillus species and various strains within a particular species, Lactobacillus reuteri, have quite different phenotypes, at least with respect to two basic phylogenetic clades. And these can be divided phylogenetically into several clades. This organism is present in a variety of other mammals, different clades. But these are the two prominent human clades. And we see one clade, clade 2, with an ability to generate relatively large amounts of histamine, shown in orange, and to suppress pro-inflammatory cytokines such as TNF using human cell culture models. In contrast, though we have a couple of exceptions, we have a very different phylogenetic clade 6, which lacks the ability to suppress pro-inflammatory cytokines such as TNF and generates little to no histamine. And when we've examined these clades, we note that the genes involved in histidine to histamine conversion are absent in clade 6 and present in clade 2. So we can explain the molecular mechanisms, partly due to the work of Pira Hemorrhagita, who's currently a fellow here at UCLA, and I understand will be promoted to clinical instructor in July. Congratulations, P. He, as a graduate student and then as a fellow in our laboratory, contributed greatly to our understanding of histidine decarboxylation in bacterial cells, the regulation of these key genes, HDCA encoding the histidine decarboxylase in gut microbes, and HDCP, a closely linked gene that encodes the transporter enabling histidine to enter the bacterial cell and histamine to exit the bacterial cell. And that provided us with a molecular explanation of how the gut microbiome may be taking dietary protein, amino acids derived from proteins such as L-histidine, to convert L-histidine into a bioactive signal such as histamine, and then to begin to understand how histamine may communicate and bind to specific receptors and suppress MAP kinase signaling. Work that was initially performed by another student in the laboratory, Carissa Thomas, currently at the University of Colorado. So we've extended these studies to try and understand how this may work in vivo. And Chunshu Gao, who completed his Ph.D. last year and has continued as a postdoctoral fellow in our laboratory, has worked on this with other members of the laboratory. These studies were recently published in MBio in December, and we've been able to show that the presence of dietary histidine in the amino acid-defined diet was important in enabling the Lactobacillus reuteri, histamine-generating human microbe in the mouse to suppress inflammation, as you see here. By gross scoring, by a Wallace score, we've shown the same thing by histology using the Amiho scoring method, and by a quantitative biomarker, serum amyloid A protein. You see a suppression of inflammation, again using this blood biomarker of mucosal inflammation, well accepted in mouse models, showing suppression with the histamine-generating Lactobacillus reuteri in blue, added to an SPF mouse model with a complex gut microbiome. We see the same effect with an amino acid diet. We lose that effect. It becomes insignificant with a histidine-free diet. So it appears that the amino acid is important in enabling the gut microbiome to convert histidine to histamine and then have a measurable biological effect, the suppression of gut inflammation. Now, I may be jumping a little ahead in the story, and so I now need to pause for a moment and describe this relatively old field that we're rediscovering in new ways in the gut, and that is histamine and inflammation, histamine and histamine receptors. We know histamine is certainly a contributor to human disease in children and adults with respect to allergic inflammation, and that allergic inflammation is working primarily through the histamine 1 and somewhat through the histamine 4 receptor. These are considered to be pro-inflammatory signaling pathways. These are G protein coupled receptors that have been well characterized for a number of years. Histamine 1 receptor goes back to the 1970s in the pharmaceutical industry as they were developing antihistamines as very profitable medications for allergy. And so it's important to note that the signaling pathway downstream of the H1 receptor is fundamentally different from the H2 receptor. We have PKC ultimately turned on by diacylglycerol, and then turning on and stimulation of NF-kappa B signaling and pro-inflammatory cytokines. Very different effect when histamine, the same molecule, binds to the H2 receptor. We have, instead of PKC, turning on a protein kinase A and a suppression of ERK phosphorylation and MAP kinase signaling, suppression of inflammation. H3 is primarily on neuronal cells, and so histamine is considered to be a neurotransmitter, and it's important as we think about the gut-brain axis to keep in mind that histaminergic neurons certainly have been well documented in the central nervous system. But our main puzzle was trying to understand how bacterial or microbiome-mediated generation of histamine may contribute to suppression of inflammation in the context of IBD. And so using the same mouse colitis model, we then added histamine receptor blockers. And so instead of manipulating the diet, in this case we used this BALB/c mouse model that Chen Xu had developed in the laboratory to compare whether we could see an effect by blocking the H2 receptor, any difference between blocking H2 receptors versus H1 receptors. We knew from prior literature that both receptors were found on gut epithelial cells. So we had a possible mechanism for signaling, and we found that only the H2 receptor blocker, specifically ranitidine, could remove the effect on, with respect to suppression of inflammation, as you see in the middle here with the Wallace score, consistent findings with SAA, H2 receptor blocker in the far right, and next to it, H1 receptor blocker, pyrilamine. Given orally, we maintain the suppressive effect with the H1 receptor blocker, but remove that effect with the H2 receptor blocker. So now we were onto something, and again, this was included in that recent publication in MBio. And we were stimulated to pursue these studies as we think about medical applications, because already in the pediatrics literature, we knew that the administration of H2 receptor antagonists were associated with severe necrotizing enterocolitis in infants, neonates. A prospective cohort of 274 very low birth weights infants showed a 6.6-fold greater risk of NEC when taking H2 receptor blockers. This has led to certainly plenty of discussion with our neonatology colleagues and colleagues in pediatrics around the administration of H2 receptor blockers and how this may exacerbate gut inflammation. So we need to rethink the administration of certain medications that, of course, are being used thinking that we might be ameliorating the problem of gastric acid production and reflux, but perhaps we're doing more harm than good. And another study published in 2012 about the same time by Josh Korzenek, who's in Boston, in collaboration with Canadian colleagues in British Columbia, using their very extensive database of inflammatory bowel disease adult patients, they found that H2 receptor antagonist exposure in adults with Crohn's disease doubled the risk of hospitalization or surgery. And you see that here in this part of the slide. In contrast, they did not see such an effect with ulcerative colitis. Now, this point may point the way towards targeted microbiome-mediated interventions in the setting of Crohn's disease versus UC. What about inflammation-associated colorectal cancer? The very last segment of this talk, which is just a follow-through of what I've been describing as you see this space-filling model of the biogenic amine histamine and its chemical structure. Could we begin to link the two? Well, we were fortunate to be in touch with Tim Wong at Columbia University and his group as they had developed the histidine decarboxylase deficient mouse. Of course, mammalian cells produce histamine. They have histidine decarboxylase in mammalian cells, including human cells. And so we consider histamine a co-metabolite produced by mammals and microbes. And in the HDC knockout mouse model, they found that using chemical agents, azoxymethane as an initiator and 3 cycles of dextrane sodium sulfate, they found that mice, HDC knockout mice, were predisposed to inflammation-associated carcinogenesis. That is, high levels of pro-inflammatory cytokines such as IL-6, as shown in the bottom right, and large numbers of tumors in the colon. And you see that nicely here. Bottom, HDC knockout mice. Above that, that's the wild type, same mouse. Both BALB/c C57 showed similar results with the same chemical agent exposure. So this was intriguing in light of our data with lactobacillus and the ability to suppress inflammation with histamine. We then slightly modified Tim's work using the same HDC knockout mouse model that he generously shared with us in moving the mice, some of the colony from New York City to Houston. We then were able to reestablish a colony and demonstrate in this 27-week study following AOM injection IP and DSS given orally, following the mice with regular administration of histamine-generating Lactobacillus reuteri, human microbe, mouse gut, and 4 different groups with negative and positive controls, and an HDC knockout strain of the bacterium that was isogenic. And what we found, and we're very close to submitting this for review, and is that we could administer histamine-generating Lactobacillus reuteri orally and suppress colonic carcinogenesis in the same mouse model by simply providing histamine through the gut lumen in mice that were histidine decarboxylase deficient, as shown here in the title of the slide. By contrast with simply media, we saw the very same phenotype of colonic carcinogenesis in these mice, more dramatically in male mice. And interestingly, when we knocked out or deleted the HDCA gene encoding histidine decarboxylase, the same Lactobacillus reuteri strain could not suppress colonic carcinogenesis. So perhaps microbiome-generated histamine could replace the endogenous histamine produced in the mouse and suppress colonic cancer. And so we've added cytokine studies, and we have more data that I'm not showing, but will be included in this manuscript, showing changes in mucosal gene expression and in this case, plasma cytokines using multiplex assays with the Luminex and Magpix platforms showing KC, the mouse analog of IL-8, greater in affected mice and suppressed in histamine generated with histamine generating bacteria in the gut. We see the same suppressive effect with respect to IL-22, also known to promote cancer cell invasion and colon cancer stemness, and I'm showing those references below the graphs. And finally, in the far right, IL-6 suppressed and statistically significant suppression of IL-6 in the plasma of these animals, seeing histamine from the gut lumen, lacking histamine in the endogenous histamine in the mouse. And we know that high levels of plasma cytokines such as IL-6 have been documented in colorectal cancer, specifically in humans. So this has been an interesting story, and we are, thanks to the support of the National Cancer Institute, we've been following this by adding histidine 2 receptor blockers, in this case cimetidine instead of ranitidine, and we're showing beginning to show increased mortality and tumor burden in an APC min mouse model. And now with female mice, we can show this effect even more dramatically in the colon and the cecum. These are tumor numbers. In the presence of the H2 receptor blocker, we see more tumors. We see increased mortality as well. And so this is an ongoing story we're continuing to add, but we've now been able to repeat this twice with 2 sets of APC min mice. And we've now, as I get to the conclusion, we've tapped this ProgGene version 2 database, initially developed at Indiana University and Purdue as a collaboration to take advantage of computational resources, combining various studies published with respect to gene expression profiling in human cancers, a variety of human cancers, large sets of human genes characterized, and we could now examine whether the increased expression of histamine receptor 2 may be protective in the context of colorectal cancer. And that's what I'm showing here. So just looking at the colorectal cancer cases in this ProgGene database and then examining the presence of elevated amounts of histamine receptor 2, HRH2, we see basically increased overall survival with HRH2 being enhanced, and we see the opposite with HRH1. So I'm just showing the high versus low, red versus green, high HRH1, lower overall survival, high HRH2, increased overall survival with colorectal cancer. So we're gaining insights as we're beginning to take lessons from the gut microbiome and applying this hopefully to a deeper understanding of mechanisms of carcinogenesis. As we begin to link mouse biology and human biology, perhaps we'll now be able to address specific receptors and signaling pathways to develop newer therapeutic interventions. And this is really exciting to us because we may be able now to focus on particular microbial genes, such as histidine decarboxylase, and pathways related to histamine generation in this case, as microbial biomarkers to stratify patients who may be at increased or decreased risk, and then to take that knowledge to begin to target specifically histamine receptor 2. The problem with histamine is that it's stimulating both receptors, but we know, and we have our hands on specific agonists that may target HRH2 and not HRH1, and that may point the way towards new interventions that may be used in combination to prevent or treat colorectal cancer, to prevent or treat patients with inflammatory bowel disease. So it's time to wrap up and to maybe share just a few general points, and that is, I started this hour talking about chronic intestinal infections or recurrent C. difficile as a primary example. And it seems clear that when we have a drastic depletion and frankly a train wreck of a gut microbiome that's been radically altered by the presence of a pathogen such as C. difficile, that simply flushing and replacing or revamping that microbiome by combinations of microbes demonstrated crudely by the application of FMT across the United States. In this case, we can see real success with FMT and taking a radical approach to the microbiome to address a very radical depletion of microbial composition and function. It works then in that context, but as we've shown to ourselves, this strategy is failing with respect to other GI disorders. And so in ulcerative colitis, FMT has been, frankly, a failure. And we've now stopped that trial, and we're now really thinking in a different way about chronic disease, chronic inflammation, colorectal cancer, other GI disease phenotypes such as irritable bowel syndrome that may require strategies that are really targeting specific mechanisms and functions. We talked about the application of low FODMAP diets in IBS and how we can think more specifically about carbohydrate metabolism, microbial composition, and response to dietary intervention. In the context of IBS, we discussed in that vein responders that may be identified by matching diet and microbial function. And finally, with respect to the histidine-histamine story, perhaps we can gain some insight that is certainly not limited to histamine, but hopefully will enable us and stimulate us to pursue additional pathways within the gut microbiome to identify new ways of stratifying patients and developing new therapies for suppression of inflammation, prevention of cancer, and beginning to link perhaps diet and microbial composition in new ways to target receptors and specific receptors such as HRH2 and those signaling pathways to develop new therapeutic strategies based at least partly on microbiome science. I need to highlight the fact that in Houston in November we'll have the 6th International Human Microbiome Consortium Congress entitled Frontiers of Microbiome Science and Metagenomic Medicine. The abstract deadline will be published very soon, sometime this summer. We'd love to see some of you in Houston in November. I need to thank the various lab members, particularly Carissa Thomas and Chinshu Gao, as well as Pira Hemarajita for the work that they've done in the lab, a variety of collaborators with respect to mouse models at MIT, where I did my postdoctoral studies, and Jim Fox's group, and our colleagues at BioGaia in Sweden, and nearby here in the City of Hope, a longstanding collaboration with Marcus Calcum and his team with respect to microbial metabolomics. And the support we've gotten particularly from the NIH, NIDDK, and NCI in particular. Various other collaborators in the Texas Medical Center, and certainly patients and families who have enrolled in these clinical studies, particularly our studies with respect to low FODMAP and irritable bowel syndrome. Thank you very much for your attention.